Hi all, welcome to lectures on microbiology. Today we're going to discuss kind of the members that make up the microbiology or microbial world, and kind of how they're classified basically. So when we look at microbiology, a lot of times people will often use the term microbes, and when the term microbes is used, then that means things that are living and non-living. Okay? If the term microorganism is used, organism suggests that the entity is alive. Okay? So when we use the term microorganism, then we can talk about two basic cell structures. We have cell structures for prokaryotes, or prokaryotic cells, and those are organisms that belong to bacteria, the domain bacteria, and the domain archaea. So those are the prokaryotes. Okay, they're unicellular organisms. Remember, the kingdom for both of these it's bacteria kingdom also, and the archaea kingdom. Although sometimes the bacteria kingdom is called eubacteria or eubacteria, meaning true bacteria, but often you'll just see it as the bacteria kingdom also. Okay, so prokaryotes, what's special about them? They do not have nuclei or nucleus, and they do have DNA. They have typically a single chromosome that's in kind of a loop or a circular chromosome. They're not linear chromosomes, so they're attached end to end, and they occur in a region within the prokaryote, which we call the nucleoid region. Okay, but there's no membrane around them, so they're floating around in the cytoplasm. That nuclear DNA or RNA is floating around in that cytoplasm. So no organelles, no membrane-bound compartments. They do have compartments. Okay, typically these are protein-based compartments, and these compartments can be utilized for lots of different things: storage of salts and maybe even storage of lipids or things that can be broken down later. But they're not organelles. They're not membrane-bound compartments. Okay. They have a single chromosome. I kind of already mentioned that. It's typically circular, or it's non-linear. Let's just put it that way. Okay, we call it circular. It's attached. They're typically fairly small. Okay? So about 0.1 to 5 micrometers in size, and that makes up the prokaryotic version of microorganisms. So when we look at the microbial world, if we look at this kind of chart here, if it's microorganisms, then it means they're alive or classified as living, so they're all organisms. If it's microbia or microbes, it might be considering everything on this chart. Okay, so the microbial world is everything, or microbes is everything on this chart, including viruses, viroids, pyrons, things that are non-living, which we just normally call acellular or non-cellular infectious agents or infectious entities, I guess. Okay. The eukaryotic version of the microorganism comes in quite a few different aspects. Like eukarya, most of you know they have a true nucleus. Okay, that's what kind of makes them eukaryotes, is that nucleus. They also have membrane-bound organelles, and I know some of you know that red blood cells in mammals, most mammals, do not have a nucleus. Okay, that doesn't mean that they're not nucleated. They're nucleated when they're like baby cells. Okay, so they lose the nucleus as they age, but they still have a nucleus. Okay? Membrane-bound organelles, so these little compartments that might have different purposes, like, in plants you might have chloroplasts and other compartments like mitochondria. In animals we have mitochondria also, like plants, and fungi have mitochondria, and protozoans will often have a form of mitochondria, or at least a organelle that potentially could be similar to that. And then, like, just all types of organelles. You have organelles that package proteins. You have organelles that break things down. So you have Golgi apparatuses, lysosomes, all kinds of things that can go on. But the reason why these structures inside eukaryotes is so specialized is they actually have a membrane around them. Okay, and that's where we get that membrane-bound organelle saying that. And typically, eukaryotes are much larger than the prokaryotes. Okay? And we'll look at a lot more differences and similarities when we look at specific organisms. But we can chart this out. Okay? We can look at the 3 domains. Remember I said the domains are kind of the highest classification. And so we can look at bacteria, archaea, and eukarya. We can look at their cell type. We know that prokaryotes, non-nucleus, no organelles, and then the eukaryotes, true nucleus and membrane-bound organelles. Number of cells. Bacteria and archaea are unicellular, although they can form large colonies and share information, share genetic information, share energy, share stimuli, but they're still considered unicellular because they can live by themselves as a single cell. Okay, eukarya, they can be unicellular or multicellular. So we'll look at unicellular protozoans, and then we'll look at multicellular organisms like tapeworms and things like that in the class. Membrane-bound organelles, like I said before, none for bacteria and archaea, yes for eukarya. Ribosomal RNA is kind of neat because this is what gives us the evidence on when these groups divided and which group is more closely related to eukarya, to us, and which group is further away, and what those differences might be. So all 3 groups, bacteria, archaea, and eukarya, have their own ribosomal RNA, and it's somewhat unique to that group. They share similarities, but that gives us this evidence of, like, when was their last common ancestor and things like that. It really helps with the evolution and figuring out kind of the tree of life and what split off first, etc. So archaea and bacteria both have cell walls. It just happens to be that bacteria have peptoglycan in their cell wall, and archaea do not. Eukarya do have cell walls, some of them, like fungi and plants, but they do not have peptoglycan in their cell walls. And there you have it. We already talked about the size of the organism. So let's look at how we classify things, just to kind of give you a little bit of the nomenclature and how we break it down. So again, we're starting off, and because microbiology, we pretty much deal with every kingdom except for plants. It is possible for us to jump from domain to kingdom, and then you're just in a whole different type of classification. And so it's kind of important to know where you might be connecting things and how to classify those things. So the domains, pretty simple. There's 3 of them. We've talked about them before. Kingdoms, we talked about it a couple lectures ago, that I typically teach a 6-kingdom kind of classification. Again, other professors teach other things. Other researchers would argue that I'm wrong. But 6 kingdoms works, and until, it's determined that it doesn't work. After kingdom is phyla, and so I'm not going to go through how many different phylum or phyla there are in the world, but just in bacteria, we know there's over 1,300 phyla in bacteria. We're just going to kind of go through a classification of a single organism now. Proteobacteria is the phyla for E. coli. Okay, and so we might look at that and say, all right, well, what do proteobacteria have in common? And then classify everything in that phyla under there. Okay, the class for E. coli is gamma proteobacteria. Okay, again, there's lots of classes of bacteria, lots of classes of other organisms, lots of orders. Enterobacterialis is the order of E. coli. Enterobacteriaceae, okay, often ACA or some form of ACA indicates that it's family. Most things that have ACA, it's just an indicator that that's the family name. So keep that in mind when you're classifying things. And then the genus Escherichia, which we normally just abbreviate as E, and then this species is Cola. So when we look at species, and we kind of talked about this at a few lectures ago, when we talk about classifying all the species on the planet, and that's a goal of a lot of individuals that study classification, is they name things. They want to find things, new things, and name them and classify them. Some of the lower estimates is that there's about 6 million species on the planet, and then again, some of the higher estimates. I mean, this is a really high estimate, but there's a few trillion species on the planet, and when it's said and done, I think bacteria and archaea will be probably the most species rich groups, and that shouldn't be a surprise to very many people when we start talking about the evolution of these species. They can evolve extremely fast, and so when we're talking hours for E. coli to replicate, and we're talking animals, you're talking weeks for the most quick animals, potentially weeks, maybe 2 weeks for like C. elegans, probably 2 weeks, something like that. That's really, really fast. But E. coli, we're talking hours. So speed and the fact that most animals have way more than a single chromosome. And so, again, who knows, right? We haven't got to that point, but we do know that they're species rich in the bacteria and archaea, and especially with new research that archaea is way more species rich than we once thought. So why is this important? Well, it's somewhat important because microbiologists tend to take things like E. coli and do like phylogenetic trees to see where different E. coli might be related to each other. And so, like studies like what kind of E. coli do we find in wastewater, human blood, livestock, meat products, and then are there connections? Is there transfer? Is there genetic transfer? Are there certain strains? So this is a single species but multiple strains, which we'll kind of talk about a lot in microbiology, is that the prokaryotes, the bacteria and archaea, they have a single chromosome, and that's what we're basically distinguishing them as a species off of. But then they have these little chunks of DNA throughout their cytoplasm, which we call plasmids, and those little chunks of DNA is enough variation that it can allow them an advantage in other environments. It can allow them antibiotic resistance and whatnot. And so we often classify, especially bacteria, as, yes, this is the species, but it's got a different type or a different variety, more so than other organisms. Okay? Just for binomial system nomenclature kind of characteristics, the genus is always capitalized, the species is never capitalized, and that you can abbreviate the genus, like in this case E. coli. It's always italicized. If you're typing it out, if you're writing it out, handwriting it out, it's underlined. Okay? So, a couple other things about the naming process and the names of organisms. Normally it honors or reflects some characteristic of the organism. For example, E. coli is honoring Theodore Rich, and you know, he's the individual that's credited with finding the species or the genera, and so it's not kosher for individuals who find it to name it after themselves. Someone else has to name it after you. So often that individual is dead and then they change the name, or sometimes biologists will get together and if you found the genera, someone else says, hey, we should name it after so-and-so, because they figured it out and they didn't name it after themselves. That's why we often say it honors so-and-so, and because they did the work, they figured it out, they found it. They didn't name it after themselves. They're not conceited like that. And then, again, characteristics. Sometimes the species name, like in this case, coli, indicates that it's colon. Like, that's where you find it. That's where the bacteria lives, which is fairly useful. Okay? A couple other things, like I just mentioned before, especially with bacteria, you're going to see that they have a strain or a type designation often, which, you know, sometimes it's based on the plasmids within the bacteria. Sometimes it's based on little bits of difference in their nuclear or their chromosome, but not enough to distinguish it as a separate species, which we can get into the species concept later. Again, informal names that resemble the genus are never italicized, so like that's why you'll see like Staphylococcus, which is the genus, and when we write it Staphylococci, we don't italicize it. Okay, so that being said, next time we meet, we're going to talk about the kingdom bacteria and start looking at some of the characteristics of just bacteria, and then we'll jump into archaea, and we'll hit some of the other kingdoms along the way. Okay? Till next time.